Okay, this is National 5 Qualifications 2026 from Qualifications Scotland. There'll be a disclaimer here saying that they own this and I don't, and I'm using it for educational purposes. And thanks to them for producing the paper. This is Biology Exam Section 2. You get your answer grid where you fill in your multiple choice question answers. And I am just going to go through this. I've already written my answers into this. I sat down and did it after the students did the exam today. I'll give you my thoughts at the end because this is pretty much the second time I've seen it. So I'm just going to go through the questions that I answered, have a look at these questions, see what we think about where the traps might be lying and where people might have come a bit unstuck. There's also a few interesting things about the paper. So let's see. They're riffing on the vacuole again. And so now we have to recognise, last year they were telling us we had to decide there was no vacuole in a bacterial cell. Now we're going to have to say that there is a vacuole, because that's definitely a vacuole in a plant cell. There is a vacuole in a fungal cell. So that's the vacuole. And the function of the vacuole in both of these cells is to store water. In plants it's for cell sap. So I'm not quite sure what answer they want there. I guess we'll have to wait and see. Okay, I would just go for storage of water. Because that's more likely to be the fungal answer. Right, but anyway, apart from the difference in size and shape, describe one other structural difference between plant and animal, typical plant and fungal cells. Well, in the plant cell we have chloroplasts, and we should remember that from our cell structure, so and none in the fungal cell. When we're doing a comparison, this is a comparison question. I'm just going to make sure I write it all out. So there are chloroplasts in the plant cell and none in the fungal cell, just to make sure that they are clear that I know what I'm talking about. Okay, select one structure from the list that is found in all typical plant, fungal, and animal and bacterial cells and give its function. Well, you don't get a nucleus in a bacterial cell, so I crossed it off. You don't get a mitochondrion in a bacterial cell, so I crossed it off. We had to choose one of these two, ribosome or cytoplasm. Easy one is ribosome, carries out protein synthesis. If you weren't sure about ribosome, you might have picked cytoplasm, which is the site of most chemical reactions in the cell. All chemical reactions in a bacterial cell, but most of them in a eukaryotic cell. Okay, let's move on. Model cell was set up as shown to investigate the movement of molecules across a selectively permeable membrane. There it is. Iodine is a small soluble molecule that changes from yellow to black in the presence of starch. Starch is a large insoluble molecule. From the information given, state the colour of the solution outside the model cell at the start of the investigation. Well, the model cell is outside the cell is iodine and water. Okay, so it's a small soluble molecule that changes from yellow to black in the presence of starch. There's no starch in that, so that has to be yellow, right? It's going to be yellow. After 1 hour, the following results were observed. The contents of the model cell turned black. So inside the cell went black. There's no change to the colour outside the model cell. So explain these results. So let's do 1. The contents of the model cell turned black. So iodine must have diffused into the model cell. It must have diffused into the model cell, and it changed the colour from yellow to black because inside that model cell, and changed colour. As you see, this is why you read through your answers again at the end of the exam. The contents of the model cell turned black because there was starch in there, and iodine stained starch black. 2, there was no change in colour outside the model cell. Starch was unable to cross the membrane. It was only found inside the model cell. Now, starch is insoluble, right? They say it's a large insoluble molecule. You might have to put in that it was a large insoluble molecule. I don't know. You're getting a mark for that and a mark from that. So really, do we have to say why it couldn't cross the membrane? You might want to say because it was too large a molecule, maybe. Don't know. We'll have to wait and see the marking instructions whether... Do we have to say why? It was only found inside. I might just say why. So this is why you read through your questions again. So because molecule insoluble and large. So I would be going through deciding between those. I think I'll just put that in to be on the safe side, because when we have to explain something, we have to give a reason, right? So why did it not cross out? Okay, but anyway. The reason why the color, there's no color change outside the model cell is because starch couldn't cross the membrane. That's the reason. So that should be the answer. Anyway, name the process that would allow water to move across the membrane of the model cell. Remember, water only moves by one way in biology, and that's osmosis. if because we're not going to have any proteins in this model cell doing active transport. There's no ATP in that system, so it all has to be osmosis, and as water moves by osmosis. And name one component of a cell membrane: phospholipid. You could also say proteins in National 5. That's the other thing we talk about. Okay, let's move on to question 3. Diagram represents a section of DNA. Give the term used to describe the structure of the DNA molecule: double helix. There you go. Watson, Crick, those guys. Name base 1. Right, here's base 1 here. You have to look for it in here. And it's complementary paired up with C. So C complementary, the complement to C is G, so it's guanine. It says name it, so we write out the whole thing. I think you're going to have to write out the whole thing. If you put G there, not sure you're going to get the mark. The base sequence represents 1.4% of the total number of bases in a single strand of DNA, right? So there's a couple of ways you can, so let's see, there it is there. Calculate the total number of bases in this strand. So there's a couple of ways you can do it. You can do it by a percentage calculation, which is that one, or you can just say that 7 equals 1.4%. So 7 divided by 1.4 is going to equal 1%. Then you can multiply that by 100. And, 7 divided by 1.4 is going to equal 1%. So 5 equals 1%. And multiply that by 100, you're going to get 500 bases. So I think that's the answer. If you do it that way, you'll get the same answer. Give the term used to describe a section of DNA that codes for a protein as a gene. They were a bit cross last year that a lot of people didn't get that, so they've asked it again. This is what happens. Questions that students find hard. It's well worth reading these course reports. Mutation occurred in this section of DNA. Explain why this mutation would result in a change to the protein's shape and function. Okay, well, it might result, but anyway, let's leave that to one side. A change in the code sequence in the gene may or will result in a change in amino acid sequence in the protein, because the code sequence determines the amino acid sequence. So that's your reason, and hopefully that'll get you the mark. Will, may, I don't think it matters at National 5, because we're not talking about different types of mutations and silent mutations and things like that. But anyway. Let's move on. An experiment was carried out into the effect of increasing pH and the activity of the enzyme amylase. Starch is broken down by amylase to produce maltose. 3 petri dishes of starch agar were set up. Each had 4 wells filled with amylase solution at the pH value shown. So there's your starch agar. So agar mixed with starch. Dump some pH 7 in there, pH 7, 10 in there, 7 in there, 3 in there, and 5 in there. And there's going to be amylase in all 4 of those wells. This is left 30 degrees C for 24 hours. That sounds like an optimum temperature for amylase. So we can't have temperature. I just realized that. They've just said the dishes were left at 30 degrees C for 24 hours. State 2 other variables not already mentioned that should be controlled to ensure the validity of this experiment. And temperature has been mentioned, so I put in temperature because I hadn't spotted that. You see, that's why you go back over your paper. So what are we going to put in? One of the variables is concentration of the amylase solution, okay? Concentration of the amylase solution. And then what do we need? No, I'm stuck. 3 petri dishes, so we need to keep constant. So the starch agar has to be the same in each. Starch agar concentration has to be the same in all 3 dishes as well. Okay, so the concentration of the amylase solution has to be the same in all of these wells. And so that should be starch agar concentration, concentration of amylase solution. Those will work, I'm sure. To explain why 3 petri dishes of starch agar were set up, that is to improve reliability. To improve reliability. Okay, don't put validity in there. It's the other one, right? It's reliability. Okay, after 24 hours, iodine solution was poured over the surface of each dish. When iodine is added to starch, it turns black in colour. We've already heard that story. Clear areas where the iodine did not turn black were found around each well, and their diameters were measured. So this is where the amylase is broken down the starch to maltose, right? So results are shown in the table. There they are. I just completed the table. It says here, complete the table by calculating the average diameter of the clear area for pH. Add those numbers up and divide by 3. There you get that. Do it twice to make sure you get the same answer twice, yeah? If you're punching, easy to make a mistake when you punch in numbers into a calculator. Quite easy to add that number in by mistake too if you're not careful. Okay, explain why there was a clear area around each well to measure. So amylase had broken down the starch around each well, right? And it broke it down around all of them. pH 3, it was not very much, but it was around all of them. Give a suitable conclusion for this experiment. Okay. I think you have to go back to the aim. The experiment was carried out into the effect of increasing pH on the activity of enzyme amylase, right? Now, you could say, I suppose, that the optimum pH for amylase is around pH 7. That would be a conclusion, right? Because at pH 7, we got the highest activity. A little bit lower at pH 10 and lower at pH 5. So pH 7, but that may not be the precise optimum pH. So you could just go for the increase-decrease structure, which makes it a bit difficult because it increases 6 to 21 to 29, and then it decreases again. So you have to say as pH increased, the amylase activity increased to pH 7. It then decreased, okay, as pH continued to rise, right? So you're going to have to put something in like that to get the marks, I think, maybe. Or you might be able to just say that's the optimum pH, but I'm not sure you're going to get away with that. We'll see. We'll see when the marking instructions come out. An investigation was carried out to compare the breakdown of glucose by different respiratory pathways in cells. The results are shown in the table. Here we go. Carbon dioxide, water, ethanol, lactate. So that one had carbon dioxide, water, no ethanol, no lactate, right? B had carbon dioxide. This is what was produced, the product. Yeah, so the produced carbon dioxide produced ethanol, and this one here produced just lactate. Okay. Use these results to identify the type of respiration in pathway A. Well, it produced carbon dioxide and water, then it has to be aerobic respiration. Totally broken down. Name a type of cell in which the respiratory pathway B would occur. B is producing carbon dioxide and ethanol. Carbon dioxide and ethanol. So it could be a plant cell or it could be a fungal cell, right? Because that's fermentation in fungi or plants. And your final one is state the location. Now, they did this last year. First here they said name a type of cell, and name a thing, and then do something different. So you just got to be careful. State the location, because you could easily just go along and say pathway C, and you could say, oh, that's an animal cell, right? But it's not. So you're going to have to state the location in the cell where that process takes place. And this is fermentation in animal cells, and it takes place in the cytoplasm, right? A lot of people, I suspect, are going to put animal in there and not get the mark. Further investigation into the energy released by these respiratory pathways followed the following results. A 16.8, 0.8. Calculate the simple whole number ratio for these results. Well, I just divided all three numbers by 0.8 and got a simple whole number ratio. 1, 1, 21. That was easy enough. No thinking to be done there. I guess they decided that because there was it went to the first decimal place that they were going to make it easy. I don't know. Anyway, handed on a plate there. Diagram shows some stages in the development of a variety of cells in the body. Stem cell, bing. Process X gets you 2 more cells. And then process Y gets you all of these cells. How exciting. Right. So, name the feature of stem cells that gives them the potential to become many different body cells. Well, they're unspecialized. Now, there are other terms that you might have learned if you've done a bit of reading ahead into higher biology. So we get multipotent and totipotent and that kind of stuff, pluripotent. But if you mention any of those, you'd probably get the mark as well, I think, from a good marker. But unspecialized is what we pretty much learn in National 5. They're unspecialized and they can still divide. Give a function of stem cells. Now, that was a tricky one, but basically they divide to provide growth and they divide to repair tissues. So I thought that it has to be one of those two, or either or, I guess, growth and repair of tissues. So that's their function, is to increase the cells, the specialized cells in the tissue and repair tissue or grow. Okay, I think that's the answer. It's going to be one of those two, right? I don't think it's going to be... I don't know, to proliferate and build an entire person, because that's what they do eventually, but I think it's a bit too complicated. Anyway, process X allows the number of stem cells to increase. name this process. Process X, 1 stem cell to 2 stem cells. How do you do that? You do that by cell division. We know one type of cell division in National 5 is called mitosis. And there we go. Stem cells can be grown in the laboratory. They double in number every 48 hours. Calculate the time taken for 16 stem cells to be produced from 1 stem cell. Okay, so how many times do you do that? So the first division you get 2, second division you get 4, third division you get 8, and the fourth division you get 16. So there's 4 divisions. And so 1 to 2 is going to be 48 hours. 2 to 4 is another 48 hours. 4 to 8 is another 48 hours. 8 to 16 is another 48 hours. So it's basically 48 multiplied by 4. It should be 192 hours. There's your answer. Name the system in the human body of which white blood cells are part of. Name the system in the human body of which white blood cells are part of. That's terrible grammar. Qualifications Scotland. Of which white blood cells are part. Right, there we go. Okay, so they're part of 2 systems, really. So they're part of the circulatory system because they're in the blood, but they're also part of the immune system. And I'm going for immune system because the white blood cells, we have learned, are fighting diseases. They fight pathogens with your phagocytes and your lymphocytes. So I'm going to go for immune system, but you might get a mark for circulatory. We'll have to wait for the marking instructions to find out. Okay. What have we got here? Diagram represents part of the knee-jerk reflex arc. Knee-jerk reaction. Identify neuron A. Neuron A, let's see. This is a rather complicated looking... so this is coming into the spinal cord, so it has to be the sensory neuron. And the one coming out of the spinal cord has to be the motor neuron going to the muscles to create the effector, or to cause the muscle to twitch. So there's no relay neuron in here. Anyway, so it's the motor neuron. Is A, because it's coming out of the spinal cord, the signal. Describe how the message travels along a neuron. Along a neuron, it's an electrical impulse, and it's transferred between neurons as chemical signals. Chemical messages you can get away with. You don't get away with chemical messenger because they reserve that word for hormones. So chemical signals are just chemical. You might just get the mark for chemical and electrical. Not sure. We'll have to wait for the marking instructions. Typical knee-jerk response takes 15 milliseconds. This is interesting. You needed to know a bit of physics here because you need to be able to calculate speed. So remember, velocity is distance over time. Let's have a look. Calculate the speed of response of the distance if the distance covered is 1.2 meters. Okay, 1 second is 1,000 milliseconds. Well, the first thing we'll do is we need to have the answer in meters per second. So the first thing we did was calculate what 15 milliseconds is in seconds. It's 0.015, right? So then we took the distance, 1.2 meters. So to calculate speed, velocity equals distance over time, or speed equals distance over time. So you put your distance on the top, you put your time on the bottom, you calculate it's 80 meters per second. It's pretty quick. 80 meters per second is rapid, okay? State the function of a reflex: to rapidly move body out of danger to protect it from harm, okay? In general, I mean, there's different kinds of reflexes, but that'll get you the mark, I'm pretty sure. Not much to write on this for quite a large diagram. Anyway, let's have a look. The diagram shows the human female reproductive system, right? Which letter in the diagram shows the site of gamete production? Female gamete production, that's eggs, the ova, and that is in the ovary, and R is in ovary. There it is. That's the uterus, this is the oviduct, and that's the vagina. Okay, so a hormone produced in the brain triggers the release of this gamete. Describe how this hormone travels from the brain to the female reproductive system. Hormones are messengers that are carried by the blood. Chemical messengers in the blood, remember that? Via the blood. Okay. Ay, caramba. So another graph. They've gone on a bit of a graph riff in this exam this year, I think. The graph shows the changes in concentration of this hormone during 1 month. There you go. There's a month, 30 days. Some months of 31, some of 28, but anyway, they've gone for 30. Concentration of hormone and the days, right? And they said, use the data to describe the change in the hormone concentration from day 0 to day 14. And this is what I put. I think it's probably good. Basically, you have to say it stays the same to there, then it rises to a peak of 75, then it decreases, and then it stays the same to there. So I just put the hormone remains at a level of 15 units for most of the period. drooping most of the period, except for a spike, and we use spike a lot for describing these kinds of graphs, that increases to 75 units between days 11 and 16. The peak of 75 units is at day 14. So I think I've got it covered. I should get the marks for that. That's 2 marks. And I probably have covered more than we need to. But anyway, there you go. Calculate how many times greater the hormone concentration is at day 14 compared to day 16. Day 14 was here, which is 75. Day 16 is here, which is, let's see, what's these two? 25, so 5, 10, 15. 15, so I put 75 over 15, and it's 5 times greater. 5 times greater at day 14. Okay. Pussy cat. Hair length in cats is an inherited characteristic controlled by different alleles of a gene. H represents the allele for short hair, and a big H represents the allele for short hair, and a little h represents the allele for long hair. We're back to H and H again. We had that before, didn't we? Anyway, the diagram shows the inheritance of hair length. That's right, it was hairy pea plants. The diagram shows the inheritance of hair length in cats. Right, so we've got a short hair crosses with a long hair. That's a homozygous dominant short hair and a homozygous recessive long hair. The F1 phenotype, all short hair, right? Well, that makes sense. And the F2 genotypes, H, big H, big H, big H, little h, and little h, little h. Okay, give the genotype of the cats in the F1 generation. Well, they're all going to get one big H and they're all going to get one little h. So your genotype is big H, little h, right? Explain why the F1 generation all have short hair. Well, the dominant allele is present in the genotype of all F1 individuals, right? And that will express the short hair variety because it's dominant, right? In addition to the phenotype shown above, some cats can be hairless. This is caused by a genetic mutation. So here's a third allele they're throwing into the mix here. Give the definition of the term mutation. Well, a definition is a change to the DNA sequence of a gene resulting in a new allele. Okay? There's your definition. And to be hairless, a cat must inherit the mutated allele from both parents. That means it has to be homozygous. It must be recessive, and it's homozygous. So state the term used to describe this genotype. The genotype? Am I going to have to put homozygous recessive? I'll tell you what I'm going to do. I'm going to put it in just in case. Take the term used to describe this genotype. Must get the mutated allele from both parents. If it gets an unmutated allele, it's not going to show. So it has to be homozygous recessive. I'm going to go homozygous recessive, okay? I changed my answer. That's why you read through your paper. That's why you go through it again afterwards and read all the questions again. You're given 2.5 hours. That's time to go through the paper twice. You can read all that yourselves. We've got this rather complicated table, and this is a tricky old question, I think. The group taking the protein supplement were compared to the group who did not take the protein supplement. Give the term used to describe the group who did not take the protein supplement. We call them the control. They're the people we basically did nothing to, and we use them as our baseline, so that's our control. Suggest one reason why the volunteers recording their own daily intake of protein could be described as a limitation. I think the bottom line is they may record the information inaccurately. Yeah, or they may make mistakes. That's not to say that scientists don't make mistakes as well, but people tend to be a little bit. Anyway, they may record the information inaccurately. They could do it inaccurately in all sorts of ways and for all sorts of reasons. That's the basic thing, isn't it? Okay, so hopefully that'll get the mark. We'll see. The individual with an individual with an initial knee strength of 50 kilograms took part in a similar training program. Predict the knee strength after 8 weeks if they took no protein supplements after each session. Okay, so no protein supplements, knee strength got them a 35% increase in the measurement. So all we have to do is predict we would predict they would get 35% as well, right? It's going to be an average. They're not going to get 35%, but our prediction doesn't have to be accurate. It just has to be reasonable. So 35% increase expected. So what is the increase of a 35% increase from 50 is 17.5? You add that to your starting 50, you get 67.5. So that's what I'm going to put as my prediction. Okay. Suggest why it may not be necessary to take this protein supplement, right? And look over here. So the protein supplement was supposed to increase strength, but if you look at it, it's increased muscle mass without the protein supplement. It's increased muscle thickness over the protein supplement. So you look good. but it hasn't actually increased knee strength. See that? So that's basically the tale you have to tell in this, because it's supposed to be increasing strength, if you read that bit. So I just wrote, while lean mass and thigh muscle thickness increased more with the supplement, knee strength increased more without the supplement. And it's going to be some combination of that works out. You could do those statements the other way around, as long as they make sense for what you understood. Look at this, guys. A 5-mark question. I've never seen one of those before. Have we? Have we had a 5-mark question before? I can't remember one. Anyway, there it is. But there's a lot to talk about. Describe the pathway of blood through the heart and associated blood vessels, starting at point A and finishing at point B. Well, there's the whole pathway, right? You could be putting arrows on here. They've given you a diagram. You could put the arrows on, but you're still going to have to describe it, because it says describe, not put the arrows on to show it. So you have to describe it. And what I did was I labelled each of the chambers with a number, and I've used the numerical pathway. So 1, 2, 3, up to the lungs, 4, 5, 6, and back out to the aorta. The vena cava and the aorta are already labelled. So I'm just going to name the whole lot. And if you want, here it is. Blood enters the right atrium 1 from the vena cava, A. It then enters the right ventricle, Z, which pumps it to the lungs via the pulmonary artery, 3. It returns from the lungs via the pulmonary vein, 4, into the left atrium, 5. It then enters the left ventricle, which pumps it to the body via the aorta, B. Valves. I labelled some valves up there. You see, like valves. Valves prevent backflow of blood, because that's part of the flow story, isn't it? Pathway of blood. That's part of the pathway of blood. Yeah, valves preventing backflow. So maybe you need to put that in to get the extra mark. I think that's it. Should that give me 5? If you knew your heart, you get 5 marks. Okay. Diagram shows the flow of energy through some organisms found in part of a food web in a pine forest ecosystem. Here they are, common juniper. There you go. Give the term used to describe all the organisms in this ecosystem. They are called the community. If you put population in here, you get no marks, because population refers to the organisms of one species. The community is the living organisms. Now, you might get, it doesn't say all the species. If it was all the species, you would say biodiversity. But, and you might get away with biodiversity, but community, I think, is the correct answer. We'll see when the marking instructions come out. That's what they want, I think. Complete the table using the information from this food web. Okay, so a carnivore is a red fox. He's eating red fox, or she's eating animals, right? Capercaillie, well, a bird, right? But yeah, everybody know that a capercaillie is a bird? Maybe some people didn't. Okay, you look at that. What the hell is that? You're saying to yourself. But anyway, remember, all of your producers are down in the bottom row. There won't be any producers higher up. These are all consumers. They're eating the producers, right? Capercaillie's a bird. Red fox is a carnivore. Pine marten is a omnivore, because it's eating juniper. And it's eating capercaillies. I don't think there's any other omnivores, is there? No, just the pine marten. So your answer is red. You could have had sparrowhawk here, not sparrowfox, sparrowhawk here, because it's eating the thrushes, and that's a carnivore as well. Pine marten. Okay, name the process by which the capercaillie uses energy. There you go. If you didn't know what a capercaillie was, you're in for a bit of fun in this exam. Use energy which will then be available to the pine marten. Right? How does it use energy which is available to the pine marten? So the energy that it uses for growth is the only energy that is available to the pine marten, because the pine marten eats its body, right? When it kills and eats it. So growth. Explain why the red fox population has the least energy. Well, at each level, this is what I put in. I might not be perfect here with my wording, and I might not get the full marks. 90% of the total energy is lost at each level of the food web. Right, so as you go up, whatever energy was in here, you only get 10% of it here. Whatever energy is in here, you only get 10% of that in the fox. So as energy is gone, but it's the fox population, really. So this represents the fox population, not an individual fox, but fox population, right? So there you go. Let's move on. Hey, we got a pyramid of numbers. Pyramid of numbers, remember, not energy, so pay attention to that. The diagram shows a pyramid of numbers for a food chain from a pine forest. And calculate the percentage of organisms in this chain that are consumers. And your consumers are always these guys up here. Your producers are the ones on the bottom row. Okay, so percentage of organisms, food chain, or consumers, you add those three up and they have to be a percentage of the absolute total. So it added up the consumers, it's 281 of them, 24 and 82, 165. Total consumer, the total is the consumers plus the 2529, so you add that up, and then you do your percentage and work it out. So 281 of 2810, which is this number, gets you 10%. Came out as a nice round number, didn't it? Right? Cool. Funny how that happens. Right. Fertilizers can be added to soil. This is a question where I think we have to wait for the marking instructions to be absolutely sure of the answers, because I think some things could be done better in this question. But anyway, let's see. Fertilizers can be added to soil to increase crop yield. Right, fair enough. Explain why crop yields need to be increased. Now, that's a bit of an open-ended question, right? You could say, well, because crop yields need to be increased because there's more and more humans every year and we have to keep feeding them, so we need more crops, right? So that's one argument. I don't think it's what they're after, but you could, it'd be true, but I don't think it's what they're after. Right? I think the answer they're looking for is that previous crops deplete nutrients from the soil, and those are replaced by fertilizers. Okay. But I'm not sure which of those answers is correct. It's a very vague kind of question. Explain why crop yields need to be increased. Is it because of human population increases? I don't know. We need more food. Or is it because the crop yields would not normally grow if there were no fertilizers there? I think it's that one. So I think this is the answer, but, well, we'll have to wait for the marking instructions and see what they give marks for. Excess fertilizers can leach into freshwater ecosystem, causing algal blooms. Describe the effect that an algal bloom will have on the number of bacteria and give them the reason why. They will increase the number of bacteria. Right? And the reason is algae block the light, killing organisms below. The bacteria increase by feeding on the dead organisms. So the bacteria are the detritophores feeding on those dead organisms that the algae have killed by blocking the light. Right? Explain why an algal bloom would cause a decrease in the concentration of oxygen in the water. Well, it's the bacteria that do that. The bacteria use up the oxygen as they increase in number and they continue their decomposing activities. So hopefully that's enough for the mark. And then it says, suggest an alternative that could be used to reduce the use of fertilizers. Now, here's the question. I don't know, and I have to look it up. Actually, I haven't looked it up yet. In National 5, is fertilizers, National 5 biology, is fertilizers defined as fertilizers made by industry, a chemical process? Because if you're going to talk about all fertilizers, which includes dung and ash and guano, etc., they're all fertilizers. So I think that's probably the wrong answer. You can't add dung because it's also a fertilizer. You can't add guano because it's also a fertilizer. For me, this would be wrong. But if fertilizers in National 5 is only referring to industrially produced fertilizers, then maybe these will be right. We'll have to wait and see. If you're not going to use fertilizers of any kind, you can move to new land and start up a new field, or you can slash and burn farming, right? It's great for the environment. Or you can use crop rotation, right? But as I say, down here, industrial fertilizer wasn't specified, so it seems to suggest that you can't use dung, you can't use ash. That's not an answer to the question, but we're going to have to wait and see. Right. Question 14. An experiment set up to investigate the changing temperature on the rate of photosynthesis. A lot of people have done this experiment in their for their assignment. The results are shown in the table. Bing, there's the table. Name the stage of photosynthesis that produces oxygen. Now, a lot of people might not remember this. They might just call it stage 1, but it's not called stage 1. You can call it the light reactions or the light-dependent reactions, or you can call it the energy fixation stage. I don't think you're going to get a mark for stage 1. It's a bit too vague, so bad luck. On the grid, complete the vertical axis and plot a line graph to show the effect of temperature on the volume of oxygen produced. So there's my... basically, you get your scale in here and you get your label, which is exactly the same as what's written in here. I'm not even putting in min here. I'm putting minute, the way they put minute there. So the whole of minute, I'm writing it out. So I'm just doing it exactly the way they did it up here. Okay, that's where it's exactly the same, right? I've got my numbers going up the side. Got a 0. Now there's a couple of things that are interesting about this. Plot the line. That looks good. Do not start your graph at the origin. A lot of people just automatically start from 0, but we do not have data here that gives a 0 value. And you don't have a 0 value. 0 temperature would be frozen water anyway, so you're not going to get a measurement there, I don't think. Anyway, the point is, if you haven't got the data, don't put the point in, right? You've got data for this, and you do have a 0 point for photosynthesis up here. Here at 70, right? You've got a 0 point here for 70. So you plot all those, draw your line, join it up. Don't do a curve of best fit or anything. Just join up the dots in biology. That's the way it works. Question 14. Describe the relationship between temperature and the rate of photosynthesis. Okay? And again, this is two relationships. As the temperature increases up to 30 degrees Celsius, the rate of photosynthesis increases. And then after 30 degrees Celsius, it falls to 0 at 70 degrees Celsius, right? So you got to write something that goes along those lines. As temperature increases, the rate of photosynthesis increases to a maximum of 3.8. I put the number in. At 30 degrees Celsius, as temperature increases further, the rate of photosynthesis decreases to 0 by 70 degrees C temperature, right? So something like that will get you the marks. I was doing this very quickly. That's why my writing's horrible. But a good examiner will be able to read that, right? Explain why no oxygen is produced at 70 degrees Celsius, right? Well, as with everything in biology, these reactions are controlled by enzymes. At 70 degrees Celsius, at least one of the enzymes that's critical for photosynthesis has been denatured. It's not working anymore. Therefore, photosynthesis stops, right? You need the full chain of enzymes to get photosynthesis working. So I just put photosynthesis requires enzymes. That's my explanation. At 70 degrees Celsius, and here's my reason for this, 70 degrees Celsius, at least one of the enzymes critical for photosynthesis has been denatured. But you could say the enzymes required for photosynthesis have been denatured. I don't think you have to be that specific. Okay? I think I got one question to go. Here we go. Venus flytraps, we love them. Right? Here's a Venus flytrap. Was that do you think that's a copyright picture? Okay, maybe copyrighted, so you can't see that anymore, can you, right? Yeah, two varieties are shown, shark's tooth and the southwest giant. We don't need to see the pictures. They're just flavour. The plant's traps are inherited characteristics that make each plant well suited to survive in its environment. Give a term used to describe this characteristic. That's the definition of an adaptation. Shark's tooth and southwest giant varieties can interbreed to produce fertile offspring. Give a conclusion that can be made from this information. They are the same species. During the evolution of this, name a process by which the plants best adapted to this environment survive and pass on favourably to their offspring. That's the definition of natural selection. And finally, because we're at the end of the whole thing, in an average trap, each side travels at a distance of 12 millimetres when closing, right? Calculate the time taken for the trap to close if each side closes at a speed of 40 millimetres per second, right? So again, you're doing a bit of physics here. You've got to remember again, at speed, velocity equals distance over time. You have to be able to rearrange that equation. You might remember your triangle. If you remember your triangle, that means time is distance over velocity. Then you just get your numbers. You work it out. It's 3.33 seconds. So if you knew your physics in this exam, at least how to calculate speed, if you remembered all that, because you don't get the physicists and the chemists, they all get a relationship sheet. We don't get it in biology, so you had to remember that one. You had to remember that. Surely most people can remember that from their BGE science, can't they? Because I don't think a lot of biology teachers will be talking about it. But you did do it way back in BGE science, so you should know how to do that. Yeah. Anyway, that's the end of the paper. What did I think of that? I think there's a couple of questions in there. I'm not sure of the specific answers that they want for their their mark scheme. I guess we're going to have to wait and see. But that's my thinking on the exam paper. If you think I got anything wrong or I misread any of the questions or didn't answer the questions correctly, by all means, give me a comment. Put it in the box below. And if you haven't seen section 1 yet, section 1, look up here. There'll be some kind of link. And you can go there and check that out. And if you were a bit annoyed that I'd already written the question, you can find another video on my channel which will have me in action writing answers, if that's what you really wanted. Okay? But it'll be a different exam. Ha! Thanks very much for watching. I hope that has made everybody feel better about their exam. I hope it hasn't made anybody feel worse. In the end, remember, National 5 is the beginning of your journey. And in many ways, the experience of doing it is by far the most important thing that you achieve today. And you can take that experience forward and use it to improve your results in the next exam. And certainly in hires and advanced hires if you're going on to do biology. Anyway, best of luck. Hope your results come out well. And I'll see you for the chemistry paper. I think that's our next one. All right. Bye.